Hello viewers, in the last few videos we studied about the vascular and cellular responses in acute inflammation. Today's topic is mediators of inflammation. So mediators of inflammation are substances that initiate and regulate the inflammatory response. The important mediators are vasoactiveamines, lipid products, which includes prostaglandins, Leukotrienes, cytokines including the chemokines, and products of complement activation. There are various other mediators also, like neuropeptides, platelet activating factors, kinins, products of coagulation. So what are the general properties of these mediators? So the active mediators, they are produced in response to offending stimuli or offending agent in the form of invaders like microbes, microbial products, or the substances released from the necrotic cells. So this ensures that the inflammation is triggered only when and where it is required. The mediators can be cell derived or plasma derived. They can be cell derived or plasma derived. The cell derived mediators can be sequestered in intracellular granules. For example, histamine in mast cells, or they can be synthesized de novo synthesis. So this is seen in prostaglandins, leukotrienes, cytokines. All these are synthesized de novo in response to some stimulus. Now what are the cells that produce these mediators? These are the sentinel cells, which includes the macrophages, dendritic cells, mast cells, but other cells like platelets, neutrophils, the endothelial cells, and some epithelial cells also. They can also secrete the mediators when these cells are stimulated, or these are induced to release the mediators. Now plasma derived, they are synthesized in the liver, and they are present in the circulation in inactive form. In circulation. So to become functionally active, they have to undergo proteolytic cleavage. a series of proteolytic cleavages make them functionally active. Example is the complement proteins. Complement proteins. Next property is that most mediators are short-lived. They quickly decay or they are inhibited by enzymes. They are inhibited by enzymes. Now, one mediator can stimulate the release of another mediator. Of other mediators. For example, the products of complement activation. They can stimulate the release of histamine. Similarly, tumor necrosis factor, it can act on the endothelial cells and it can stimulate the release of interleukin 1. Now these secondary mediators, they can have similar functions as the primary mediators, or they can also have opposing effects. Next we discuss in detail the vasoactive amines. The important vasoactive amines are histamine and serotonin. They are named vasoactive because they have important action on the blood vessels. They are stored as preformed molecules in cells. Coming on to histamine, the richest source of histamine is the mast cell. The mast cells are present in the connective tissue adjacent to the blood vessels. Other sources are platelets, basophils, and these the histamine is present in the mast cell in the form of granules, and they are released in response to stimuli by degranulation. So what are these stimuli that lead to release of histamine? These are physical injuries in the form of trauma, cold, heat, in response to various anaphylatoxins, C3A, C5A, these are the anaphylatoxins. And when antigen binds to IgE antibodies, which are displayed on the surface of the mast cells, this leads to the degranulation of the mast cells. And this forms the basis of the immediate hypersensitivity reaction. What is the action of histamine on blood vessels? Histamine causes vasodilation of arterioles. It causes increased permeability of venules, and this is the principal mediator that is responsible for immediate transient phase of increased vascular permeability. There is increase in the interendothelial gaps in the venules. So how it acts on the vessels? On the endothelial cells there are receptors for histamine, and histamine binds to these receptors and produces these actions. Now various antihistamine drugs are available for treatment of allergies and other inflammatory reactions. Now how they act? They act by binding to these receptors, and thus they inhibit the action of histamine. Next is the serotonin. Serotonin or 5-hydroxytryptamine. This is produced by platelets and by certain neuroendocrine cells in GIT. Serotonin acts as a neurotransmitter. It acts as a neurotransmitter. And it also produces vasoconstriction. Its role in inflammation is not clearly understood. Next we come on to the arachidonic acid metabolites. Next is the arachidonic acid metabolites. These are the lipid mediators. These play important role in the vascular and cellular events of inflammation. These are the prostaglandins, leukotrienes, so they are derived from arachidonic acid, which is present in the membrane phospholipids, Arachidonic acid is a 20-carbon polyunsaturated fatty acid in icosa tetraenoic acid. Tetraenoic, it has 4 double bonds and it is 20 carbon. Icosa in Greek means 20, and these are the position of the double bonds. So the source of arachidonic acid is the dietary source. It is derived from linoleic acid, which is essential fatty acid, and the active arachidonic acid, it is released from the membrane phospholipids by the action of the injurious stimuli and by mediators like C5A, and this is released through the action of enzyme phospholipase, or so for cellular phospholipases, particularly the enzyme phospholipase A2. Now the arachidonic acid derived mediators, these are also called as icosanoids. Icosa means 20, so they are derived from 20 carbon fatty acids. They are named so because they are derived from 20 carbon fatty acids. Now they are produced by the action of 2 enzymes, cyclooxygenase which produces prostaglandins and lipoxygenases. This produces leukotrienes and lipoxins. Now with the help of a diagram, let us understand how the arachidonic acid metabolites are produced and what are their roles in inflammation. So this diagram shows the production of arachidonic acid metabolites and their roles in inflammation. So these are the two pathways, cyclooxygenase and lipooxygenase. So from cyclooxygenase pathway, the prostaglandins are generated, and the lipooxygenase, when it acts on arachidonic acid, it leads to production of leukotrienes and lipoxins. So there are two enzymes, COX-1 and COX-2. COX-1 is constitutively expressed and it can also be induced by inflammatory stimuli, and COX-2 is expressed only in those cells which are taking part in inflammatory response. So these are the main mediators generated from this cyclooxygenase pathway: prostacyclin, thromboxane A2, PGD2, E2, PGF2 alpha, and these are produced by action of specific enzymes on the precursors of this pathway. These enzymes have limited tissue distribution, like thromboxane synthase is present in platelets, so this leads to production of thromboxane A2 in platelets, and it is a platelet aggregator. Thromboxane A2, it is a potent platelet aggregator and it leads to vasoconstriction. Whereas in the endothelial cells, the thromboxane A2 thromboxane synthase is not there, prostacyclin synthase is there. So this leads to production of prostacyclin, which has opposite action. It is it causes vasodilation and it is inhibitor of platelet aggregation. The PGD2, E2, they lead to vasodilation and increased vascular permeability, and PGD2 plays important role in chemotaxis. And these prostaglandins, they play important role in the pathogenesis of fever and pain. PGE2 is hyperalgesic. Now coming on to the leukotrienes and lipoxins. The lipooxygenase pathway leads to production of leukotrienes and lipoxins. Leukotrienes play important role in vascular and smooth muscle reaction and leukocyte recruitment, whereas the lipoxins, they suppress inflammation. There are three types of lipooxygenases, but 5-lipoxygenase is predominant in neutrophils. When it acts on the arachidonic acid, it leads to production of 5-hydroperoxy-icosa tetraenoic acid, and this leads to production of 5-hydroxy-icosa tetraenoic acid, which is a potent chemotactic agent, and this acts as a precursor for other leukotrienes. The leukotriene B4 is also a potent chemotactic agent. Leukotriene C4, D4 and E4, these are cysteine containing leukotrienes, and they lead to vasoconstriction, intense vasoconstriction, bronchospasm and increased vascular permeability. Now the bronchospasm and increase in vascular permeability is even more than that produced by histamine. The lipoxins are suppressors of inflammation. They inhibit neutrophil adhesion and chemotaxis. They require two cell population for their biosynthesis. In the neutrophils, the precursor forms are synthesized and these are then passed on to platelets, where the active or mature lipoxins are synthesized. Now coming on to the pharmacologic inhibitors of prostaglandins and leukotrienes. Corticosteroids are broad spectrum anti-inflammatory agents that reduce the transcription of genes that encode proteins which are involved in inflammatory response. For example, the enzyme phospholipase A2, COX-2, the inducible nitric oxide synthase, and several cytokines, pro-inflammatory cytokines like interleukin-1 and tumor necrosis factor. Now there are cyclooxygenase inhibitors or non-steroidal anti-inflammatory drugs like aspirin, indomethacin. So they act by inhibiting the enzyme cyclooxygenase. Aspirin irreversibly acetylates and inhibits cyclooxygenase, and this leads to prevention of production of prostaglandins. So prostaglandins are not produced. So this explains why they reduce fever and pain, because prostaglandins play important role in the pathogenesis of pain and fever. There are specific COX-2 inhibitors which are inhibiting only the action of COX-2, which is taking part in the inflammatory response, whereas the action of COX-1 remains intact. So there is production of prostaglandins, which are cytoprotective, and this reduces the side effects of gastric ulceration, which is seen with the use of non-specific cyclooxygenase inhibitors. But the specific COX-2 inhibitors, they increase the risk of cerebrovascular events because they are inhibiting the production of prostacyclin in the endothelium. Prostacyclin is a vasodilator and inhibitor of platelet aggregation, while maintaining the production of thromboxane A2. This action is COX-1 based. So thromboxane A2 remains, it continues to be produced, so it leads to vasoconstriction and it promotes the platelet aggregation. So there is a increased risk of cerebrovascular events with the use of COX-2 inhibitors. Now other anti-inflammatory drugs are 5-lipoxygenase inhibitor, the leukotriene receptor antagonists. So these are used in asthma because they relieve the bronchospasm that is caused by the action of leukotrienes. So this was about the mediators of inflammation. In this video we studied the vasoactiveamines, the arachidonic acid metabolites. In the next video we will be studying about the complement system, the cytokines and other mediators of inflammation. I hope you like the video. For more such videos, please subscribe my channel and do not forget to like and share my videos. Thanks for watching.